This method can help answer key questions in the field of host-pathogen interactions, such as what are the unique characteristics of the productively infected cells, what host cofactors allow viruses to establish productive infection, and how to target these cells in therapeutic interventions.
The main advantage of this technique is that it provides quantitative measures for both protein and mRNA within single cells, and most reagents are commercially available.
Demonstrating the cell sorting procedure will be Matthew Creegan, a senior technician from the flow cytometry core in Dr. Michael Eller's laboratory.
To begin, in a designated pre-PCR molecular biology workstation, prepare the assay mix by combining 96 gene expression assays into an RNase-DNase-free tube, making sure to add each assay to a final concentration of 180 nanomolar of forward and reverse primers. Add DNA suspension buffer to achieve the appropriate dilution of the assay mix. For each anticipated 96 by 96 chip array, pipette 6 microliters of each assay into a designated well of a 96 well PCR plate. Then seal the plate with an adhesive. To begin surface staining viable cells in a tissue culture biosafety cabinet, first prepare the compensation samples and the master mix of fluorescent antibody cocktail as outlined in the text protocol. Thaw the cryopreserved cells in a 37 degrees Celsius water bath for 2 minutes. After this, add between 0.5 and 2 milliliters of the cell suspension to a 15 milliliter tube.